Okay, so in lab today, we're going to talk about the eye. And I want to start off by talking about the structures that are surrounding the eye or attached to the eye. We refer to those as the accessory structures. So we have muscles, and then we have some structures with some hair on it, and then some structures that produce liquid, and then structures that cover stuff. So let's run through each one of these here so we can get a better appreciation of everything that's involved in our visual apparatus here. So let's start off with the eyebrows, okay? I used to think the eyebrows were something that I would just raise when someone said something ridiculous to me, and I would just say, what the heck's up with that? Or when you're looking at Dwayne "The Rock" Johnson, and he'll give the people's eyebrow. Well, they're really important, especially living down here in the South, for preventing sweat from getting into your eyes. And for those of you that have worked outside in the middle of the summer, played any activities, ran, whatever, and you're sweating a lot, the eyebrows help to prevent sweat from getting into your eyes and sometimes stinging those eyes. The eyelashes are great for removing anything or preventing stuff. If it's a windy day, especially down here in Greenville, South Carolina, we have pollen that likes to float around, that green stuff that we see covering our cars in the springtime. Right. Well, the eyelashes help to prevent things from actually making any contact with your eye. And what will happen is if an object brushes your eyelash there, it will initiate the blink reflex. And we learned about the sensory receptors that are associated with some of our hair follicles. Those are called hair root plexes, and they wrap around the follicle of the plexes and they monitor displacement of the hair. Well, if something strikes one of your eyelashes, guess what? Boom, you'll stimulate that reflex because of those hair root plexes. Right? And we will find the eyelashes on our upper eyelid, the superior palpebrae, and the lower eyelid, the inferior palpebrae. Speaking of your eyelids, here we are. We call those palpebrae. You have a superior palpebrae and an inferior palpebrae. And we'll see not only are the eyelids just a flap of skin, but there is a muscle inside, and we learned about it way back in chapter 11, the orbicularis oculi muscle. We've seen it contract when you're squinting your eyes. And then something to give your eyelids a little bit of support, all right, is the tarsal plate. That's the fibrous core at the center of the eyelid. And so there's a muscle that helps you when you want to open up your upper eyelid there. It's called the levator palpebrae superioris. I really like the name of this muscle because it tells you exactly what it does. It elevates the upper eyelid, levator palpebrae superioris. So basically what it's going to do is it's going to widen what we call the palpebral fissure. And that's that opening, that eye slit in between, all right, the superior palpebra and the inferior palpebra. Now both the inferior and superior palpebra will meet up with each other. And they'll join together on the lateral part of your eye and the medial part of your eye. Where it meets up on the medial part, we call that the medial palpebral commissure or canthi. Where they meet up on the lateral portion of the eye, we call that the lateral palpebral commissure. And if you'll notice on the medial, you can even look in the mirror. If you'll notice next time, you'll see this pink thing near your nose, right? Near the medial palpebral commissure there. And that's called the lacrimal caruncle. You'll notice some of you might have this issue when you wake up in the morning and you feel some solid material in that area. Some people, we call it in my house eye boogers or eye boogies, right? Well, the lacrimal caruncle, a lot of times, anything that does get into the surface there of your eye all right, the tears or the lacrimal fluid will push that stuff off to the lacrimal carbuncle there. In our tarsal plate, right, and we'll go over this when we're doing some labeling here later on, we have these glands. And there's a series of glands. There's a whole bunch of glands associated with your eye. This is the first group of glands that we're going to talk about tonight, the tarsal glands. And they produce, all right, those are sebaceous glands. And if you recall, sebaceous glands are going to form a more of an oily type of secretion, something that's more viscous than our merocrine glands, which are our sweat glands, because they produce more of a watery secretion. Well, the tarsal glands produce, all right, this oily secretion here. And again, a lot of it helps to lubricate and nourish and protect the eye itself. So when these glands get infected, we call that a chalazion. I like saying that, chalazion. And so an infection of those glands is the chalazion, whereas you might notice some folks say, you know, if you look at somebody and they have like a red bump near the margin there of the eyelid there, all right, sometimes close to one of your eyelashes, we refer to that as a stye. That's an infection of the sebaceous and sweat glands that are near the eyelid base there. All right, so let's talk about now the covering of parts of your eye and parts of your eyelids. And that's called the conjunctiva. Some people pronounce it the conjunctiva, either or, whatever floats your boat. But it's a transparent covering made up of our stratified columnar epithelium. And we call that specialized because it's transparent. And so there's two locations for the conjunctiva. All right, the part that covers the sclera, which is the white part of your eye, that's the ocular conjunctiva. And then the part of the conjunctiva that covers the internal surface of the eyelid, the underside of your eyelid, that's going to be referred to as the palpebral conjunctiva. Well, both of those conjunctival liners will meet up with one another and they'll form the conjunctival fornix, which is basically where they just both come together and connect. In this structure, in this lining, we are going to see our unicellular goblet cells, and they're one of the more abundant type of glandular cells, and their job is going to keep the eye from drying out. All right, that's very bad. We do not need that to happen. All right, there are parts of your eye that do not have blood vessels, right? So they have to rely on diffusion and other active transport processes to get nourishment and get rid of wastes. So these goblet cells help to keep the eye moist. We'll also see in the conjunctiva, all right, blood vessels. We know what their job is to do. And then, of course, we'll have a series of nerve endings for nervous innervation. Now, what we will see, and this is important to note, that the conjunctiva does not cover the cornea. The cornea, right, is that transparent structure that covers both the pupil and it covers the iris. I'll show you on a picture here. If you're not quite sure, I'll show you all this stuff. So it does not cover the cornea, so therefore it is not going to have any role in light entering the eye. The conjunctiva plays no role in that. The cornea does. Remember what I said, all right? There's that concept that when light travels through different media, right, structures with different densities, it will bend the light, right? Well, the conjunctiva does not play a role in any light refraction. The cornea does, but not the conjunctiva. And of course, if you get an irritation to this structure, we call it conjunctivitis, also known as pink eye. All right, so let's just quickly talk about some of these structures here. You heard me talking about them. Let me show them to you. All right, so here at the top, we can see there's the eyebrow. And then we have our eyelids. Here's the superior palpebra. Here's the inferior palpebra. Then you have the eyelids coming off the margins here. right, so this opening here is the palpebral fissure. So when your eyes are closed, obviously it's more like a slit. Now when they're open, it's much wider. All right, here you can see the pupil, that black part there, and then surrounding the pupil is the colored portion of your eye, and that is the iris. The white here of the eye, that is the sclera. And then the conjunctiva covers the sclera, but not this part here. The cornea is a transparent structure that covers over the iris and the pupil. So here's the cornea would be here. The conjunctiva, the ocular conjunctiva would be over here. Where we see the upper eyelid and the lower eyelid meeting up with one another on the lateral portion, that's called the lateral palpebral commissure or canthus. And then here medially where they meet up is the medial palpebral commissure or canthus. And then this little pink fella right here, that's the lacrimal caruncle, and that's where you'll get a collection of goo or gunk here in the morning when you get those eye boogers there. All right. So here's a nice sagittal cut through the eye. And we can see here, we're looking at the superior palpebra and the inferior palpebra. All right, here's the orbicularis oculi muscle, obviously covered by skin. And then here you can see the tarsal plates. That's that fibrous core structure. And then you'll have the tarsal glands here by that tarsal plate there that will secrete that sebaceous fluid there. All right, here's a nice picture of the cornea, nice transparent structure. No conjunctiva covering the cornea. Then immediately behind the cornea, you have the iris, colored part, and then the opening there is the pupil. Right behind the pupil is our lens. All right, we talked about it a little bit in lecture. You can see the suspensory ligaments that are attached onto the lens, and then our ciliary muscle up here. Right, so you can see here on the white of your eye, the covering, there is the palpebral conjunctiva, and then, the ocular conjunctiva, and then the conjunctiva will kind of fold back on itself and attach here onto the palpebra. All right, and that's the palpebral conjunctiva. And where they both kind of meet, that is the fornix there, the conjunctival fornix. I think I hit everything. All right, yep, good. Okay, so let's continue on our mission and our journey of talking about the accessory structures of the eye here. And I want to talk about the lacrimal apparatus. It's kind of a cool scenario. I'm going to explain to you why when you get a good cry going on, like a really good cry, not an ugly cry, you know what I'm talking about, because there's some people that when they cry, man, whoo, you'd rather not see that image. You want to scrub it from your brain, okay? But a good cry, I'm going to tell you why your nose gets runny when you cry. So to do that, we're going to talk about the lacrimal apparatus and then talk about its function and its purpose here. All right, the lacrimal apparatus, its job is to produce lacrimal fluid and then collect the fluid and distribute it across the eye, and then to drain the fluid away from the eye. So what's in lacrimal fluid? Well, the majority of it, just like with a lot of things in our body, is water, right? We also have, all right, sodium content there, right, which contributes to some, you've probably heard the terms that salty tears, right? Well, for it to be salty, you're going to need some chloride there, all right? But the sodium helps to give it that close to salty taste sensation, right? We have lysozyme and antibodies. The last two are for our immune system, okay? Antibodies, we know, are proteins that help to fight off primarily viral infections, but they can kill off other things too. And then we have our lysozyme that helps to kill off bacteria. So the purpose of lacrimal fluid is to lubricate the eye, clean it and keep it moist, keep it from drying out, and it helps to reduce any friction from the eyelids. All right. We also get some help from those tarsal glands too. that help to reduce some of that friction. So there's a lot of things at play here, all right, to really kind of keep your eyes in good working order. And then, of course, we have the antibodies and the lysozyme that help to defend against microbes. And then we need to provide nourishment to the cornea, which is that clear covering over the iris and the pupil. But more importantly, right, because it does not have a direct blood supply, we need to oxygenate the cornea. We need to provide plenty of oxygen, right, for its metabolic processes. So where does this whole apparatus start? It starts with the lacrimal gland. Lacrimal gland is located in the superior lateral portion of the orbit there, closer to the ears. And so it's what is going to produce our lacrimal fluid. And so to disperse the fluid, all you have to do is blink your eyes, and it starts to move that lacrimal fluid across your eye. Roughly, it's about 15 to 20 times per minute that you blink. And I'm sure just me mentioning blinking, some of you or most of you have probably blinked a couple times. But that's what happens, right, with the lacrimal fluid. Now, eventually, we've got to get rid of this lacrimal fluid. So as you're blinking and it's flushing across your eye, your fluid is going to move from lateral portion of your eye, from where it was produced by the lacrimal gland, towards the medial portion where it's going to get drained out of. So next time you're brushing your teeth, look at yourself in the mirror and pull down your lower eyelid and look towards the medial portion of your inferior palpebrae. You'll see a little black hole there, close to the lacrimal caruncle. That is called a lacrimal puncta. The puncta is the opening. You have one on your superior palpebra, your upper eyelid, and you have one on your lower eyelid, the inferior palpebra. And those holes, right, are going to drain into the pipe system of your lacrimal apparatus to drain the fluid away from your eye, right? Those holes are the opening to what we call the lacrimal cannuliculus, or just the lacrimal canals. So you have a superior lacrimal cannuliculus and you have an inferior lacrimal cannuliculus. Both of those, all right, lacrimal cannuliculi will drain, they'll fuse and drain into, not fuse, but they'll both drain into the lacrimal sac. And the lacrimal sac then is going to take that lacrimal fluid and dump it into this structure called the nasal lacrimal duct. And this duct connects the lacrimal apparatus to your nasal cavity. So then your lacrimal fluid will mix with any of the mucus that's located in the nasal cavity. Hopefully it's not a lot, but I'm sure living here in Greenville and in the seasonal the seasonal period there for allergies, you might have a little bit more mucus, but what will happen is lacrimal fluid gets mixed with the mucus and you'll swallow it. So now you know why when you have a good cry and you start to tear up, all right, whatever tears don't run down your cheek are going to be flushed into the lacrimal puncta, into those lacrimal cannuliculi, then into the lacrimal sac, then into the nasal lacrimal duct, and then into your nasal cavity. And that's why you get a runny nose. So that's what the tears are. There's just excess lacrimal fluid. So here's a great picture here to show you what it was I was just describing. All right, so here's the lateral portion of your eye. Here's your lacrimal gland sitting up here in the superior lateral portion of the orbit. And it's producing the lacrimal fluid. So as you blink on and off, that circulates the fluid, okay, throughout the eye, and it works its way towards the medial portion of your eye. And then you can see these openings here in each of your eyelids, right? You have, those are called the lacrimal puncta, and they are the openings to your superior and your inferior lacrimal cannuliculi. And both of those drain into the lacrimal sac here. And then that lacrimal fluid will drain inferiorly down into the nasal lacrimal duct, eventually into the nasal cavity, where you'll swallow it or it'll run out of your nose or you'll spit it out. So that is how tears happen and work. So now you know. And so you can explain that to all your friends, and then everybody will sing your praises. Okay, so let's actually get right into the eye. All right, we're done with the accessory structures. Now we want to learn about all these different components to the eye. The eye is probably one of the more complicated sensory organs in our body. And I will not be doing enough justice in this time that I have, but I hope you can get a little bit of an appreciation for it as we move through. Okay, so if you thought your eye was spherical, it's not. All right, it's close, but it's not completely spherical. And folks that have astigmatism, those eyes are even less spherical than a regularly shaped eye. All right, so obviously we have two eyes, one in each orbit. Like, you know, we have a right and left orbits, which are the holes in your skull. And your eyes, I want you to keep in mind, your body it's wonderful in how it's been set up and designed and whatnot. And so your eyes, when they sit in the orbit, they're not just sitting there and just attaching to muscles and just kind of banging around. They're surrounded by periorbital or orbital fat. And as you know, one of the purposes for fat is insulation. Another one is energy. But in this scenario, it's for cushioning. Okay, helps to fill that space like packing material, and it holds, because that's what loose connective tissue is. All right, our loose connective tissue makes great packing material. Well, fat does the job. So when we look at our eye, we're going to divide our eye into two cavities. You have a posterior cavity, which is the larger of the two cavities, and that's behind the lens. Our lens is going to be our reference point here, okay? And then the cavity in front of the lens is the anterior cavity. Okay, so behind the lens, posterior cavity, in front of the lens, anterior cavity. So we have some substances that occupy these cavities. The one in the posterior cavity is called the vitreous humor. And we describe it as the permanent vitreous humor because during fetal development, it is produced and it doesn't go anywhere. Okay, it's going to stay with you. All right, whereas when we talk about the substance in the anterior cavity, all right, that is called the aqueous humor. And we describe it as the circulating aqueous humor because your eye is constantly producing it and circulating it around the anterior cavity, and then it drains off. Similar to like cerebral spinal fluid. All right. You have the choroid plexus up there in the brain, and they're producing the cerebral spinal fluid, and it circulates around, but then eventually, you know, we get rid of it as we're making more. All right, so that's what we're going to see here with the aqueous humor. Now, the vitreous humor is more like gelatin. Okay, it's going to, or jello or pudding. Okay, so it's going to have a little bit more of a thicker consistency. The aqueous humor will be closer to water's consistency, not quite as the same consistency as water, but close to it. So when we're looking at our eye, we have three layers. We call them tunics. You have the outermost layer, which is the fibrous tunic. The middle layer is the vascular, and then our inner layer is the retina. Right? Those are our three tunics. We're going to go through all of those here. So here you can see a picture here of your each eyeball and the orbits they live in. And you'll notice here's the optic nerve coming off the posterior part of the eye. That is cranial nerve number 2. Cranial nerve number 2. And you can see here, we are highlighting the fovea centralis. That's that area on your eye that has very sharp vision. And that's where the majority of your cones will be located. Those photoreceptors, those cones that are for color and sharp vision. And you'll be utilizing those in, again, I don't want to say bright light, but like daylight conditions, lit conditions. All right, so here you can see the three layers, the three tunics of the eye. And so we should tell you what are found in those tunics. The fibrous tunic is the outermost. There's the sclera and the cornea. Okay, the sclera is the white of your eye. The cornea is that transparent structure. We'll talk about that in a second. That goes over your pupil and iris. Then we have our middle layer, which is the vascular tunic. The iris is part of the middle layer, the vascular tunic. We also have this structure called the ciliary body. And then you'll have this other layered structure that goes all around the eye there, and that's called the choroid. And then our innermost layer is the retina. And it's made up of two layers, the pigmented layer, which we talked about. We saw a little picture of the pigmented layer. That's the layer where the photoreceptors are embedded into. The outer segment of those photoreceptors are embedded into the pigmented layer. Also, the pigmented layer is where our transretinol, remember that molecule that gets straightened out during the bleaching reaction in our rods? Well, that's where that transretinol wanders off in shame, all right, once it becomes disassociated from the opsin. And so that's where it meets up with ATP, and that ATP is going to bend that transretinol back into the cisretinol configuration. All that happens in the pigmented layer. And we'll talk about what happens in the neural layer in more detail. We've already kind of talked about it. We were talking about the phototransduction. All right, so if we get a little bit more of a detailed picture here of our eye, I'm not going to go through everything, but I do feel like I need to go over a few things here. So a couple of the big things. Here's the lens. Remember, this is our reference point. So everything in front of the lens is going to be part of the anterior cavity. Then everything behind the lens is the posterior cavity. This is where the vitreous humor is going to be located. And then your aqueous humor is going to be located up through here. Right, so we can see on the lens, we have these little structures here, these white structures. Those are those suspensory ligaments that are attached onto the ciliary muscle here. And we learned when the ciliary muscle contracts, that causes these ligaments then to slacken and become loose, right? And then we'll see, all right, the lens, all right, fatten up or thicken. Now, when the ciliary muscles relax, that causes the suspensory ligaments to tighten up, and that causes a lengthening or thinning of the lens. All right, a couple other things here. You can see the iris, the opening here in the iris is the pupil. All right, up here, you'll notice there's a space here in front of the lens or the pupil here, or excuse me, not pupil, the iris, up to the cornea. And that is the anterior chamber. So we divide our anterior cavity into two compartments or chambers. So if we're in front of the iris, it's the anterior chamber. If we're behind the iris, okay, but still between the lens and the iris here, we have our posterior chamber. Now that will become relative or important here when I start talking about how the aqueous humor starts to circulate throughout here. You'll see this little hole down here. That's the scleral venous sinus. That's the drainage point for the aqueous humor. So the aqueous humor gets produced back here. It circulates all around and it gets drained out of this little structure here. If something happens to this hole, all right, you can get a condition called glaucoma. We'll talk about that in a little bit. All right, that's good. Oral serrata, I'll talk about that, but the oral serrata is this jagged edge structure here, right? And that's going to be found closer to the anterior part of the posterior cavity. All right, back here, you'll notice there's this little indentation here. That's the fovea centralis. That's where our sharp vision occurs. And then this structure right here is called the optic disc. The optic disc is where blood vessels enter and exit the eye, and where the ganglion cells, their axons, all right, will begin to form our optic nerve back here. There are no photoreceptors here in the optic disc, none, okay? All right, I think that's good enough for that. And we'll just go on to the specific structures of the fibrous tunic. All right, so the fibrous tunic is that tough outer layer. All right, and when you look at the term fibrous, you should think, all right, this is going to be made up of, all right, a dense irregular connective tissue that will give support and structure to the actual eyeball itself. So our fibrous tunic is made up of the sclera and the cornea. And the sclera plays a huge role in giving us the shape of the eye. And what we'll see is because of it's made up of the dense irregular connective tissue, all right, and think about it. Something else that's made up of dense irregular connective tissue is the periosteum on your bone. And what connects to the periosteum of your bone? Muscles. Well, here on the sclera, we have our extrinsic eye muscles that attach onto it. Like I said, dense irregular connective tissue is a great connective tissue. It can handle multi-directional stresses, which is perfect for the extrinsic eye muscles or almost any kind of muscle. All right, the other structure is the cornea. This is important. The cornea, nicknamed the window, is a convex, transparent structure. All right, it allows the passage of light into the eye. All right, interesting fact, though, it is made up of epithelial cells, specifically, all right, squamous epithelium. All right, and what do we know about epithelium? It's avascular. All right, so this structure has no blood vessels. All right, but it is, excuse me, a layered structure. The outermost layer is made up of stratified squamous. The inner layer is simple squamous, and then it's like a sandwich. In the middle, we put a layer of collagen in there. But the whole time, this whole structure is transparent, so it doesn't interfere with light entering into the eye. The only way that it, how do I want to say this, interacts with light is that it refracts the light. It is going to start the bending or the refraction of the light because, again, we're trying to focus that light onto the fovea centralis. So that's where the first bit of refraction starts in the cornea. All right, the second layer, excuse me, is the vascular tunic. And so obviously when you look at the name, all right, vascular, you should be thinking vessels. Well, I think blood vessels initially, all right, but we're not just going to limit it to blood vessels. We're going to include our lymphatic vessels. And lymphatic vessels play a role, all right, in immune function, all right, but also they help with extracellular fluid drainage too. And we'll also see in this layer what we call our intrinsic muscles. And we're going to talk about them here in a few moments. So the three parts to the vascular tunic are going to be the iris, all right, the ciliary body, which is going to contain those ciliary muscles that we saw that help to manipulate the shape of the lens, and then the choroid. And that's that kind of pinkish structure that we were looking at that goes and surrounds the posterior portion, excuse me, the posterior portion of the eyeball. So let's start off with the choroid first. All right, the choroid is going to have tons of blood vessels, capillaries, all right, because that's the smallest type of blood vessel there. And so its job, as you know, capillaries, all right, I don't know if I've mentioned this before, but when you think of capillaries, and if you have me for 211, I will definitely remind you of this. Think of capillaries as the exchange site between your blood, your cardiovascular system, and the tissues. This is where your blood dumps off oxygen, for example, and other nutrients to the cells, and where it picks up carbon dioxide and cellular wastes. So there's a lot of exchanging going on, all right, in the capillaries. So that's what's happening here. Also, we'll see in the choroid how there's the presence of these melanocytes, and we know the job of melanocytes. They're going to make that melanin, that dark that dark colored substance that helps to protect the nuclei in our cells. But in this scenario, all right, the melanin is going to absorb any light that's just kind of bouncing around back there, right, in the eye, which is good, right? Because again, we don't need to have excess light just hanging out, right? Because it can interfere with our visual acuity. So the next structure in the vascular tunic is the ciliary body. And so there's two parts to the ciliary body: the ciliary muscles, which we've talked about, and then the ciliary processes. So we're going to see the ciliary body just in front of the choroid. So if I go back to this picture here, here is the choroid right here, and then our ciliary body, all right, is going to be here. Okay, so it's just anterior, just in front of the choroid there. So again, ciliary muscles are going to connect to the lens through the suspensory ligaments. And so when those muscles, this is, you need to memorize this. I hate to say it. I wish there was an easy way to explain it. All right, when the muscle contracts, it loosens the suspensory ligaments. Right, and so when we loosen the ligaments, we're going to change the shape of the lens. We'll talk about it here in a little bit. And then our ciliary processes, we haven't mentioned this, right, contain tons of capillaries, and they're going to make the aqueous humor. Similar to how the choroid plexus and those ependymal cells of the choroid plexus made the cerebral spinal fluid, we're going to see something similar to that. Okay, these capillaries here, all right, that are located in the ciliary process are going to help produce that aqueous humor that's going to circulate throughout the anterior cavity, both in the posterior and the anterior chambers there of the anterior cavity. And then finally, the last structure here of the vascular tunic is the iris. Now, most of us know the iris as the colored part of your eye, okay? And so it does play an important role because we have smooth muscle in the iris. And we've talked about it back in chapter 15 when we had parasympathetic and sympathetic innervation to the muscles in the iris will either constrict the pupil or dilate the pupil. Along with the smooth muscle in the iris, we'll see more melanocytes. And of course, we know what their job is. It's going to be to, well, in this case, the melanocytes in the iris are what give the iris your eye color. Same thing in your hair. Melanocytes give your hair its color. And then also we'll see more vessels, blood vessels and lymphatic vessels. And then we'll also see some of our neuronal structures in there. So the iris is going to be our reference point when we're talking about the anterior cavity. The anterior cavity is divided into two parts. So if we are in front of the iris and we go all the way up to the cornea, that's the anterior chamber. If we go back from the iris to the lens, we're in the posterior chamber. That opening then in the center of the iris is the pupil, and we can actually affect the size of the pupil utilizing two different types of muscles. When I say types, two different named types of muscles. It's smooth muscle. The first one is the sphincter pupillae muscle, and this is the muscle that is going to cause constriction of the pupil. It is stimulated by the cranial nerve number 3, which is ocular motor, and that is part of your parasympathetic nervous system. Remember the cranial sacral division, right? You had 4 cranial nerves, cranial nerve 3, 7, 9, and 10, right? So when this muscle occurs, and I'll explain it to you, it's part of the parasympathetic nervous system when this muscle contracts and it constricts the pupil. The other muscle in the iris is called the dilator pupillae muscle. I love the name of these muscles because they tell you exactly what they do and where. All right, so the dilator pupillae muscle is going to cause dilation of the pupil. It's innervated by the sympathetic nervous system. So here you're seeing, all right, two different parts or divisions of the autonomic nervous system innervating the eye. which brings me to the pupillary reflex. You've gone to the doctor, they've taken a pen light and they shine it in your eye. And what they're doing is they're seeing if your pupils constrict. So they're checking how well your cranial nerve number 2 is working. Remember, reflexes have a motor component and a sensory component. Back in chapter 14, when we were learning about those different types of spinal reflexes, for example, the stretch reflex, the sensory neuron was transmitting that stretch sensory input to the spinal cord, and then the spinal cord would then activate motor neurons to cause the muscle to contract, right? Well, here, all right, when we're talking about the pupillary reflex, the sensory component for this reflex is the optic nerve. Optic nerve is sensory only. So that light information gets transmitted into the optic nerve. And then when the muscle, pupil contracts, right, we are then stimulating the sphincter pupillae muscle. And that, the nerve that controls that muscle is cranial nerve number 3. So cranial nerve number 2 and 3 have to do with the pupillary reflex. And that is based upon how bright something is being shown in your eye. So here you can see, right, we've shown the light into this person's eye, and we'll see how the pupil gets smaller. And the reason why is you have this sphincter-like muscle. And we learn in chapter 11 that any sphincter-like muscle is always going to make the aperture or whatever opening that it surrounds smaller. And so that's what we see here. The sphincter pupillae muscle, which is this little grouping of muscle here, right, it starts to contract, making the pupil smaller. That is through parasympathetic innervation. Now, at night, if you were to go outside, like if you're taking your garbage can out to the curb and it's dark, right, your pupil will get really big and it dilates. And the reason why is it wants to try to get as much light into your eye to try to get as good of an image as possible, whatever you're looking at. So the dilator pupillae muscle contracts. That's this grouping, that's all this. All this out here is the dilator pupillae muscle. And you can see, right, when it contracts, these muscle fibers here on the radial portion become smaller because they're contracting, and that makes the opening, the pupil, larger. That's under sympathetic innervation. Sympathetic innervation. Okay, let's get into the inner layer, the retina, also known as the neural tunic. This is where all that phototransduction and all that fun stuff occurs here. So we have a couple components here to the retina, the pigmented layer, the neural layer, and then the ora serrata, which is that jagged edge there, right? And then we'll see, oh, I'm jumping ahead here. We'll get back to that. Let's quickly go over the pigmented layer. This is that layer that is going to be deep to the choroid. But this is where we're going to have vitamin A. So when you eat that carrot that your mom gives you because she says, hey, carrots are good for your vision, all right? When you break it down, you have that beta carotene in the carrot, and we release the retinol from that, the vitamin A. It travels into the pigmented layer, all right, amongst other places in your body, and it is going to provide, all right, that retinol for our photoreceptors. The pigmented layer, because of the melanin or the melanocytes, again, will help to collect any stray light. So it's not interfering with our vision. The neural layer, all right, the neural layer, this is where we find our photoreceptors. All right, so our rods and cones will be here in the neural layer, and we'll talk about those other cells that I didn't get a chance to finish talking about, the bipolar cells, right, and our ganglion cells, and there's a couple other here that we'll talk about here in a second. All right, so here in the neural layer, this is where we get our light energy coming in, and then we undergo phototransduction, and we convert it into our action potentials, our nerve signals there. The oral serrata, all right, that's that jagged edge structure I was telling you about. This is pretty much the cutoff point, all right, of where we have photoreceptors and no photoreceptors. It's that boundary, right? So we'll see, right, around the oral serrata, we will not have any photosensitive parts there. All right, so let's talk about, all right, the layers here in our neural layer of the retina. Remember, there's two layers. We've got the pigmented layer, and then we have our neural layer. All right, so the pigmented layer is, oh, I think I have a picture. Yes. Goody. Let me just show you real quick. Okay, so here you can see, we're showing you 2 out of the 3 layers here, or tunics, excuse me, 2 out of the 3 tunics here of the eye. This outer part here, all right, that's the fibrous tunic. And then you've got this pinkish layer here, all right, that's our vascular tunic. And then deep to that is going to be the retina. So since we're in the posterior part of the eye, all right, the vascular tunic, this is the choroid. And so we can see it at a better close-up vision. You can see, look at that, tons. Now you know why we call it the vascular layer, right? Tons of vessels all throughout. Right. So then sitting on top of that, we have our retina. And so we have the pigmented layer, very thin, okay? But that's where those photoreceptor cells kind of embedded themselves into. Then our neural layer has 3 layers to it. Okay, you've got your photoreceptor cell layer, then you've got your bipolar cell layer, and then you have your ganglion cell layer. And then in between, we've got a couple other cells. We've got horizontal cell and amacrine cells. But those guys, we'll talk about that. All right, back. There we go. Okay, so now that we've gone over that, so the 3 layers of the retina, all right, that are found in the neural layer. Okay, let me rephrase that. The retina is made up of 2 layers, the pigmented layer and the neural layer. Then we have 3 sublayers in the neural layer. Our photoreceptor layer, okay, and that's relatively easy. That's the outermost. That's that layer that's right up against the pigmented layer. That's where our rods and cones are. This is where we're going to see that phototransduction occurring. Then as we move towards the center of the eye, we have our bipolar cell layer. This is that the cell layer the photoreceptors are going to synapse on. Remember how I was telling you how with the rods, all right, at night in the dark, all right, we do depolarize the cell and it releases the neurotransmitter glutamate onto another cell. That's the bipolar cell. And as you know, the bipolar cell has a central cell body with 2 processes going off in different directions. So that's pretty much what it looks like there. Right. So the dendrite here will have processes at one end, and at the other end we'll have the axon, but at the end of the axon, it will have its synaptic vesicles and all that fun stuff, which will synapse onto the ganglion cell. All right. So the bipolar cell is going to receive any input from the photoreceptor cells, the rods and cones. Then that bipolar cell synapses onto the ganglion cell layer. That's the innermost layer. All right. After that, it's the actual cavity of the posterior cavity there. Then we're just going to have the vitreous humor there. All right. So the ganglion cell is going to receive input from the bipolar cell, and the axons of the ganglion cell will eventually, all right, form your optic nerve. And that occurs at the optic disc. Remember the optic disc is that's where the blood vessels enter and exit the actual eye, and where all the axons of the ganglion cells that will eventually form the optic nerve. So the other 2 cell types, the horizontal cells and the amacrine cells, right, they're going to help, all right, communicate, all right, between your first 2 layers of the neural layer, between the photoreceptor cells and the bipolar cells, right? So they'll coordinate information and messages between those cells. And then the amacrine cells will obviously, all right, those are found between the second and third layer of the neural layer, which are the bipolar cells and the ganglion cells. All right, so we saw that picture. All right, let's talk about parts of the retina. All right, the optic disc, we already talked about that, also known as the blind spot, no photoreceptors. All right, but that's where the ganglion axons go back and form the optic nerve. All right, now there's a new structure here called the macula luta. So when I think of the macula luta, obviously I have to think of the fovea centralis, right? Because that sits inside the macula luta. So it's like a circle within a circle. The macula luta is the bigger circle, right? And it sits lateral to the optic disc. That's important that you know that. But what's more important is that the center of it is the fovea centralis here. That's where we have all these cones located there. And that's where your sharpest vision is going to be located. That's where we're trying to get our light to shine on for whatever we're looking at. Because when you're looking at anything, what you're seeing is the reflection. Light is bouncing off whatever you're looking at. Right? I don't want to get into all the physics with you, right? But that's what you're seeing is a reflection. Light's bouncing off whatever you're looking at, and it's going into your eye, and your eye is picking that up. Right? So we want to focus everything on the fovea centralis, but sometimes that doesn't happen, and you'll see the light shining, and we'll talk about different types of vision, impaired vision descriptions here next week. But on the periphery, on the outer portion of the retina, right, that's where we're going to see our rods. So primarily in the fovea centralis is where we're going to see cones, but on the periphery, that's where we're going to see rods. And if you think about it, when you dilate your eye like this, you're going to let more light in. Well, the fovea centralis only needs a little bit, okay? But all this other area here that you're seeing the pupil, all that light will hit the peripheral portions of the retina. And usually if your eye is dilated that much, it's usually at night, or unless you're under a lot of drugs, okay? But let's not think of the drugs, right? But in a dark room or wherever, your pupil will be dilated so much, and it's trying to get as much light in, and it's going to try to get the rods to be stimulated, right? And we're going to find them more on the periphery of the retina. So we got to open that door to let as much light, even though it's a low light situation, we got to try to let as much light in so we can stimulate those rods so we can get our night vision going on. So when you're looking at this picture here, let me just zoom in. What we're showing you is light entering into the eye, and we're trying to focus it here on the fovea centralis here. So you can see, all right, here is where your optic nerve and where the blood vessels enter and exit the eye. That's the optic disc. The fovea centralis is going to be lateral to that. And here's the other fovea centralis on your right eye. It's going to be always lateral to the optic disc. So when we look at the ophthalmoscope, this is what your doctor is looking at when the doctor shines that light into your eye with that little that tool. It's called an ophthalmoscope. So what they're looking at are a couple of things. One, they're trying to look at where the optic disc is, and it's easy to find. Just trace the blood vessels back. All the blood vessels converge on the optic disc. So that's where blood vessels enter and exit the eye. And that's where all the axons from those ganglion cells will head towards this structure and then exit out of the eye as the optic nerve. So if you go lateral to that, then you'll see the macula luta, this big circle structure. And then at the center, it's a little bit of a depression. There's the fovea centralis. That's where your sharp vision is located. All right, let's get into the lens here real fast, okay? The lens is obviously going to be the structure that is going to focus light rays so you can get a clear picture. So the lens has to change. For those, it used to be much easier to describe this to folks when a lot of people used to have cameras. Now everybody has a cell phone, so they just take pictures with that. But anyways, right, when you change the shape of your lens, you are going to change the focusing of light on the retina. Remember, our goal is always to try to hit that fovea centralis. Right? So the lens is made up of, all right, of a clear, excuse me, I can't think that, of a clear protein center called crystalline. Right? And during the developmental process, when you were growing up, you lost most of the organelles for the cells within the lens. And over time, all right, there was a theory, I was telling another section about this, that you know how a tree, every year of its life, you know, if you were to cut it down and you look at the trunk, you could count the rings around the tree. Well, there used to be a theory, I don't know if they've done any further research on it, that your lens was the same way. Every year of your life, right, that your lens would put a layer, all right, of this dense fibrous elastic material. And so every year it would add another layer, another layer, another layer. But over time, your lens becomes less elastic. And so hence, that's one of the reasons why people need reading glasses, because it can't change shape as well as it used to, and it can't focus the light. So you need a little of help with eyeglasses. So the theory was that your lens would put these layers on. And when I was in med school, I remember reading about it, but I never followed up on it. And so I don't really know why I told you guys that. But anyways, if you've ever heard about it and you know of anything, let me know. I'd love to find out what the deal is. So the main purpose of the lens is here: to impact light refraction, to bend the light rays so we can get a sharp, crisp image. And to do that, right, we have to manipulate the shape of the lens. So that means we are going to involve the ciliary muscle and our suspensory ligaments. Now, I've already talked about this, so I'm just going to kind of move through this kind of quickly. All right, so I'm going to give that example. You're driving down the road, you're looking in the distance, and then you glance down at your radio. So when you are looking, all right, at something far away, 20 feet or more away, all right, the ciliary muscles are relaxed. The suspensory ligaments tense up or are taut. That causes the lens to flatten out. All right. Just the opposite happens when you look down at your radio. When you look down at your radio, you undergo accommodation. All right, the muscles now, the ciliary muscles tense. The suspensory ligaments become slack. And then the lens thickens and becomes more spherical. That's what we talked about tonight in lecture class. So now I want to talk about the different humors in our eye, because that's when I talk about the aqueous humor, that's going to lead us to glaucoma. So the vitreous humor is that permanent gelatin that sits in the posterior cavity. That's that part of the eye that sits behind the lens, right? It's permanent, does not go anywhere. And so its main job is to maintain the shape of the eye. And it also helps to support the retina by pushing it up against the other tunics. So I'm sure some of you have heard about a detached retina, and that sucks. It's not very fun. And the vitreous humor helps to prevent the retina from detaching in the fact that it helps to push it and keep it flush against the other tunics of your eye. Now, the aqueous humor, this is the one, right, that is less viscous, it's more watery, and it's going to be in the anterior cavity there. And that's going to be that cavity that's in front of the lens, but we've divided it up, the anterior cavity, into the posterior chamber and the anterior chamber. Okay, when we talk about the chambers, our reference points become the iris. So from the iris to the cornea, that's the anterior chamber. From the iris back to the lens, that's the posterior chamber. And so the aqueous humor gets produced in the posterior chamber, right, by these ciliary processes here, right? And so its job is to produce the aqueous humor. The aqueous humor is going to help, all right, with some of the nourishment of the cornea and some of the other structures there in that region. And so when we start the production of the aqueous humor, right, we have these capillaries that sit in the ciliary processes located in the posterior chamber. So aqueous humor is filtered out and it circulates from the posterior chamber into the anterior chamber. And how it gets there is that it goes through the pupil. And then it moves into the anterior chamber, circulates around there, and then eventually is drained out by the scleral venous sinus. Unfortunately, if we cannot drain that out, we are stuck with glaucoma. And that's an increased pressure in the eye. So that's what this picture here is kind of showing us here. All right, here are our ciliary processes. All right, they're producing the aqueous humor in the posterior chamber here. It circulates through the pupil into the anterior chamber here, doing its thing, nourishing all these structures here. And then it drains out here through the scleral venous sinus. Now you notice over here, this is called the angle here. And there's two types of glaucoma. There's open angle and closed angle. And that has to do with what's occurring here at the angle of the eye and what it does to occlude the scleral venous sinus. Because you block this up, the aqueous humor can't drain out. All right, quick picture here of the different muscles here in the eye. All right, we're going to talk about the extrinsic eye muscles real fast. You have got 6 of them here that are going to affect the movement of the eye. There's 4 muscles that end with rectus, and then there's 2 muscles that end with oblique. You have the superior rectus muscle, which is the muscle that attaches directly on top of the eye. You have the inferior rectus muscle that inserts directly underneath the eye. We have our lateral rectus, that's on the side closest to the ear. You can't see the medial rectus, but that's on the other side closest to the nose. Then we have our superior oblique muscle. This is the one that has that little pulley system here called the trochlea. It attaches onto the superior lateral portion of the eye. Then you've got the inferior oblique muscle that goes from the medial portion of the orbit, and then it comes out and attaches onto the lateral underside of the eye here. So that's a lateral view. Here you can see it from the medial view. Here's a nice picture of the medial rectus. Then again, you can see the superior oblique a little bit better. All right, there's that little pulley system there. All right, now this is just a picture of all the muscles without the eye. Superior rectus, medial rectus, inferior rectus, lateral rectus, superior oblique, inferior oblique. And then you can see it here from the superior point of view. Okay, let's stop here for the eye and do just a little bit of labeling real fast. All right, if you have your atlas ready, let's do it. We'll go through a couple structures here. Okay, so here you can see in an anterior view of our model, where we're looking at the upper eyelid, that is called the superior palpebra. That's your top eyelid. Then right below it, we have the inferior palpebra. And it's really nice when you're looking at the superior palpebra because you can see this muscle here that attaches onto it. We're going to label that in a second. You probably already know what that is. All right, now where the superior palpebra and the inferior palpebra meet each other on the medial portion near the nose, that's called the medial canthus or the medial commissure. then you can see the lateral canthus or the lateral commissure on the lateral portion of the eye. Again, that's just where both of your eyelids come together and join up. Here you can see the covering of the sclera, the white of your eye there, that is called the ocular conjunctiva. And then you can see the palpebral conjunctiva, that's the lining there on the inside of your eyelid there. Then you can see our tarsal glands there. Those are those lined structures right there. And those tarsal glands are going to be producing, all right, that sebaceous fluid that is going to help reduce friction, all right, help with keeping the eye from drying out. So those are the tarsal glands. This is an internal view of the palpebrae there. All right, you'll notice we got this structure in the superior lateral portion of the eye. That's the lacrimal gland. It produces the lacrimal fluid that is going to produce tears when you're crying. And you can see, all right, we have these kind of gray, you have this gray line here, but then you have these smaller gray lines coming off of this big gray line. Those are the lacrimal gland ducts. All right, the lacrimal gland ducts. And then the medial portion of our eye, you can see that pink body structure there. That's the lacrimal carbuncle. That's where your eye boogers collect. You don't have to know this for labeling purposes, but you can see we have these dark holes here. Those are the puncta. Those are the openings here, all right, that the lacrimal fluid will drain into. So the puncta are the openings to our canaliculi. So the one up top there is the superior lacrimal canal or canaliculi. And then right below it is the inferior lacrimal canaliculi or canal. And both of the canaliculi are going to drain lacrimal fluid into the lacrimal sac. The lacrimal sac sits in the lacrimal sulcus of the lacrimal bone. Wow, how many times do I have to say lacrimal? All right, so there's the lacrimal sac. Lacrimal sac drains down into the nasolacrimal duct. And from there, the lacrimal fluid drains into your nasal cavity. All right, here's that first muscle there that I was telling you about. This is the one that opens up the eyelid, the superior eyelid. That's the levator palpebrae superioris muscle. All right, it is the most superior muscle when we're talking about the muscles of your eye, not including orbicularis oris. So here you can see, this is not in your atlas, but you can see it from this angle here. Right, is medial to the lacrimal gland. It sits on top of the superior rectus muscle. Okay, and here you can see it on the lateral view here. That is the levator palpebrae superioris muscle. So right below it, this is superior rectus, right here. Okay, so the superior rectus muscle, all right, when it contracts, it causes your eyes to look upwards. Superior rectus. Then, way on the bottom, okay, you have the inferior rectus muscle. The inferior rectus muscle attaches onto the bottom portion of your eye. So when you contract that muscle, you look down. If you want to use the optic nerve as a reference point, here's the optic nerve. It's going to travel anterior and it's going to connect directly onto the back of the eye. So the superior rectus muscle is right above it. Inferior rectus muscle is right below it. Here you can see it better. There's the optic nerve going into the posterior portion of your eye. So you can kind of see the inferior rectus muscle down below it here. All right, this is the remains of superior rectus. We cut it away. All right, there's the inferior. You don't have this, so I'm going to skip through that. All right, here you can see the medial rectus muscle. This is the medial portion of the eye. Okay, you can see the medial rectus also. All right, superior oblique muscle is up in this corner here. Okay, and so the superior oblique muscle is on the medial side of the eye. So this muscle here must be medial rectus. When you contract that muscle, your eyes will turn in towards your nose. Remember we were talking about, all right, the convergence of your eyes when you're looking at something close by. This muscle is the muscle to thank for that. So when you're looking at something far away and then you look down at your radio, right, this muscle here is going to pull your eye in toward your nose, so as your eyes converge while you're looking at something close by. The lateral rectus muscle, right, easy to tell where that is because here's the lacrimal gland. The lacrimal gland's on the lateral superior portion of the eye, so it's on the same side as the lateral rectus muscle. This muscle here, you'll look out toward your ears. Right, that's the lateral rectus muscle. Here you can see the superior oblique muscle up in the top corner here. Here's the tendon for it, which will attach onto the eye here. You can see it just a little bit. There's the trochlea. Speaking of trochlea, the trochlear nerve, cranial nerve number 4, is going to be the nerve that innervates the superior oblique muscle. All right, then down below, you can see, here's our inferior oblique muscle. It starts here in the medial side of the eye, and then it attaches on the underside lateral portion here of the eye. All right, so there's this little formula over here you can see on the right side of your screen. All right, LR6SO4 over 3. And so this tells you what cranial nerves innervate what muscles. LR's lateral rectus, cranial nerve number 6, is the abducens cranial nerve. SO is superior oblique. The number 4 is the 4th cranial nerve, which is the trochlea. And then all the other muscles are innervated by cranial number 3, which is ocular motor. So that includes medial rectus, inferior rectus, superior rectus, and inferior oblique. It's just a quick way to memorize which cranial nerves innervate which extrinsic eye muscles. All right, I'm going to stop here. This is a good place to stop. This is where I stopped my other section, so I feel pretty good about that.